Well, welcome, everyone. We're so happy to have you here joining us for another science talk. One of my personal favorite things that the Noyo Center does are these science talks that we have and get together and learn about things from people from far and near and far, and bolster up our knowledge on our backyards. So whatever the case may be, I'm just happy to see that you've all joined us here tonight. I would like to introduce you to our guest speaker, Terry Gosliner, who is probably down the hall and around the corner from the team that I work closely with in the marine mammal world, Sue Pemberton and Mo Flannery. So he's in one of those hallways, and what a place. What an incredible place. If you haven't been down to California Academy of Sciences, get on down there and spend some big fat hours down in the aquarium world. And wow, we're so lucky to have that place. So Terry, I'll let you tell your story and tell us first off how you first came upon the fascination with nudibranchs, and then tell us more about yourself.
Okay, great. Thanks so much, Sarah, and thank you all for um, having me here this evening and being able to join you remotely. I'd much rather be up there right now on the beautiful Mendocino coast. But I grew up in the San Francisco Bay Area. I'm a California native, fourth generation Californian. And when I was growing up, the ocean was always part of my life, and I was I loved swimming, snorkeling, and at age 16, I became a certified scuba diver. But already, when I was in high school, I had a fascination with nudibranchs, and I had an amazing mentor, high school biology teacher who took kids out to the tide pools, and we had field trips, and that's where I saw my first live nudibranch. A lot of my work has been on the California coast, but I've also spent a lot of my career studying tropical reefs in remote places like Papua New Guinea and the Philippines and getting a taste for the diversity of the tropics. But I always love to be studying things and exploring here in our own backyard. And so that's a little bit about me. I went to, for my bachelor's, I went to the University of California at Berkeley, and then I went to the University of Hawaii for my master's degree, and then to the University of New Hampshire for my PhD. And after getting my PhD, I also uh, traveled to South Africa and worked at the South African Museum in Cape Town for 3 years, where I was exploring the unique nudibranchs of Southern Africa, which was a great opportunity. So that's a little bit about me. I'm going to start sharing my screen and tell you a little bit about what I hope to talk to you about this evening. Hmm. All right. So Um, nudibranchs are, of course, some of the most beautiful creatures that live in the ocean, and um, but they're not just a pretty face in many ways. They are sort of sentinels about changes that are going on in our world, and they provide a lot of insight into the process of evolution and how various adaptations take place. And they tell us about climate change and all of the other things that are going on in our rapidly changing world. and they provide great insights into to developing new conservation strategies. So I think that's where I want to focus is what's going on in our tide pools, how do nudibranchs shed light on that, and then hopefully end on a hopeful note and one that mobilizes people to action. So in places around the world where there's a lot of upwelling, um, you find a lot of diversity. So certainly on the California coast, the coast of Southern Africa, the west coast of Southern Africa, the west coast of South America and in Chile and Peru, in New Zealand, and then on the coast of Japan, where you also have upwelling. And these are represent the most diverse places that in terms of their temperate um, oceans. Nudibranchs in the tropics are even more diverse, but these are the richest temperate habitats around the world, and it's all driven by upwelling, um, the colder water coming to the surface and being nutrient rich and driving that entire food web. If you go back to your biology 101, uh, the nutrients are what drives primary productivity and then creates opportunities for other organisms up the food pyramid to just explore lots of opportunities that's driven by that nutrient rich water. Well, California's tide pools, as most of you know in your own backyard, are home to remarkable temperate diversity. And there are amazing creatures that live on our shores, and many of them are now undergoing a lot of threats, and we'll talk a little bit about those. But there's also some hopeful signs and things that we can all do, and things that biologists can do to, first of all, understand those changes that are taking place and to take some action, which will help hopefully turn some of the disruptions that we're seeing to our marine ecosystems around and continue to foster those really rich, diverse habitats. Well, nudibranchs are mollusks. They're marvelous mollusks, as are so many of the other organisms that you find along the California coast. And some of them are actually dominant organisms like mussels that form vast beds and provide habitat for a whole bunch of other organisms. And basically, um, we have just such a rich variety of mollusks and, you know, from the tiniest of creatures like the little snail on the right hand side that only gets up to about 10 millimeters in length to things like the red octopus and even the giant Pacific octopus, which is one of the largest octopus species that exists on the planet. Um, most of you have seen chitons in the tide pools, that these strange mollusks that have 8 plates. Most of them sit there in a little territorial um, scar that they produce by eroding the other surface of the rock and will forage around that home scar. But some of them, like this one shown here in the middle, has an extended head region. And instead of just grazing on algae and small invertebrates, this actually lifts up that head and traps crustaceans and is a carnivorous chitin. And so we're always really surprised and amazed by the diversity of adaptations that we find. And mollusks and other organisms that make up some of the dominant areas of the intertidal regions between the tides really are instrumental in shaping marine habitats. And so we'll talk a little bit more about that as well. But I want to put nudibranchs in a broader context of mollusks. Nudibranchs and their relatives are in a group called the Heterobranchia, and um, they're an unusual group of mollusks that we didn't really understand was a distinct group of things that were seemingly very disparate in terms of their morphology and their biology. And it's only through using DNA to be able to study the evolutionary relationships of these organisms did we find that these things are all closely related cousins of each other. And nudibranchs are certainly the group of these heterobranchs that has the largest diversity in marine habitats. But some of them still have a shell, others like the Sacoglossa, which are herbivores, have completely lost their shell as an adult, and nudibranchs have completely lost their shells as adults. Um, and others still have an internal shell or an external shell. And some of them, like the pteropod on the lower right, make their living by swimming around in the plankton and feeding on other snails that are found swimming in the open ocean waters of the California current, where there's a lot of nutrients in that system. So this gives you an idea of who's related to whom, and um, I'd be happy to talk to you more about that if you're interested. But that just gives you a little context of who are the close relatives of nudibranchs, and how do we understand that is largely by we've gained a lot of insights by studying the anatomy of these animals, but really by the new tools that sequencing DNA and how that allows us to reconstruct a more accurate representation of the family tree of nudibranchs and their relatives do we get all these additional insights and know who the closest relatives are. So the nudibranchs really have two fundamental groups. And the easiest ones to tell apart are the dorids, called the Doridina, and they're always characterized by having a circle of gills on the hind end of their body. And having that circle of gills is um, important in respiration, but it's also reflects their evolutionary history. And you find that within these groups that have this circle of gills around the anus of the animal, um, they're able to um, use the gill for respiration, but it also serves some other functions that we'll talk a little bit about later. And the other major group is called the cladobranchs, which we've only really figured out as a natural group. And it includes things like Melody, which lives on kelp beds and feeds on crustaceans, to things like this aelid nudibranch, Hermacenda being one of the most common ones on our California coast. And what is unique about them is that they're carnivores, like most nudibranchs, but they've specialized on cnidarians, the jellyfish, sea anemones, and hydroids and their relatives, which all defend themselves by producing stinging cells and organelles that are um, used for the defense of the sea anemone. But nudibranchs have been very clever in that they can steal these stinging cells when they ingest their um, cnidarian prey, and they can then transport those stinging cells to the tips of these finger-like projections called serrata, and then store them and use them for their own defense. So This is a really unusual case of being able to develop a defense mechanism derived strictly from their prey and utilize it for their own defense. So, um, how that actually happens, we're beginning to get an understanding of that, but it's still all the details of that relationship are still poorly understood, and there's lots of opportunities for young scientists to unlock some of the mysteries about the biology of nudibranchs. So nudibranchs feed on a wide variety of prey, and here's a couple of species of nudibranchs that we found on an expedition in the Galapagos. All of these turned out to be new species. Um, the one in the upper right is an aelid nudibranch that feeds on hydroids. It has its closest relative is found here in California, and its closest relative has a purple body, bright purple body with orange serrata, and it's called the Spanish shawl. And so this one in the upper right is still an unnamed species. The one in the lower right is a predatory species, and you can see there's another nudibranch hanging out of the mouth of that species. And so it's a predator on other species of nudibranchs. And this one we first found in the Galapagos, but it's also been found in Baja California. And so it's a predator on other nudibranchs. The one on the lower left, you might not be able to see very well because it's very cryptic on it, and it's on its natural food, which is a species of sea fan or gorgonian. And the way in which we first found this was it was on our last dive of the expedition that we went to on. I've been on several expeditions to the Galapagos, but on this particular one, we had an expert scientist from the academy who studies soft corals, and he'd forgotten to get a sample of this very common species to be able to take back and study the genetics of it to make sure that it was not a distinct species that was found outside of the Galapagos. And so he broke off a small fragment about the size of your thumb, and he brought it up, and we were looking at it in the plastic bag, and I noticed all of a sudden that there was a nudibranch that I'm circling here. Um, and this was a brand new species. We had never seen it before in the wild, that despite looking many times, and sure enough, it was a new species, and we've only seen it a handful of times since then, and it's still an unnamed species. So lots of times, discovery of new species, which we do a lot of, even on the California coast, is really serendipitous. You're just very lucky, and there just happened to be that nudibranch on that little fragment of soft coral that was plucked off of the gorgonian. So, as I mentioned, many relatives of the nudibranchs, the sacoglossans and this sea hare that is commonly found on eelgrass beds in embayments like Humboldt Bay. Um, Even though these are both herbivores, they feed it by a very different manner. The one on the right has a broad ribbon of teeth that it uses to scrape diatoms off the surface of the eelgrass that it's found upon. The one on the left, however, is a member of another group of herbivores that feed by using a single ribbon of teeth that's only one tooth wide and a long band. And it basically uses those teeth to pierce single algal cells and uses a muscular pump of its body to basically feed on the suck out the individual contents of those algal cells. So even though these are both herbivores, their adaptations and their evolution of feeding mechanisms can be very different. And most species of Dorid nudibranchs feed on sponges, and a lot of them have a ribbon of teeth that is made up of individual teeth, but is very broad and has teeth that have individual cusps all over them. Whereas this other species that feeds on bryozoans or moss animals, which are colonial encrusting animals, they have a very different radula that is more like a file that simply scrapes off the surface of the bryozoans rather than ripping off hunks of the sponge tissue that you find in the other species of dorids. So there are lots of adaptations, and these have allowed nudibranchs to undergo all of the diversification that we've seen. Aelid nudibranchs and other cladibranchs feed on cnidarians, as I mentioned, and store nematocysts in the case of aelids. And the aelids that feed on hydroids, they have a ribbon of teeth that is only 3 teeth wide, whereas this one on the right, which feeds on soft corals in the lower intertidal, has a much broader ribbon of teeth that's used to scrape off the tissue. So we find tremendous variety of forms, which is one of the reasons that we see such a diversification of nudibranchs in California, is that each one of them is doing entirely different things of utilizing different resources to be able to survive. I wanted to just give you a brief snapshot of their reproduction as well, and then we'll start talking about some of their diversity. Nudibranchs and their relatives are all hermaphroditic, meaning they have both sexes in the same individual. And here you see two nudibranchs lined up with their reproductive organs shown here on the right-hand side of the body. And it's almost like when two of them mate, both of them exchange sperm with each other, and both of them then have their eggs fertilized by the other individual. They can't fertilize themselves. They produce an egg mass often. On the lower left, you can see the ribbon of eggs that this nudibranch has produced. And those eggs then usually hatch out as a planktonic larval stage. And what's really interesting about the larval stage, um, from my perspective, is that it has a shell. It has a coiled little snail shell, and it really reflects the evolutionary history that nudibranchs have lost their shells, but in their development, they still have a larval shell. One of the exceptions to that is shown in the lower right-hand picture, where there are very yolky eggs that bypass the planktonic larval stage, and the individuals hatch out as miniature little nudibranchs without a shell. And so uh, there are some variations that are species specific. Each species has a different developmental mode and different adaptations to living not only in its environment as an adult, but in terms of their larval biology and the way they can make it through their dispersal phase, which is the larval stage that usually has a shell. All right, so we've learned a lot about the diversity of California nudibranch species. And if we go back to when the first nudibranchs were discovered on the California coast, which is in the mid 1700s, and it was largely the Russian explorers that um, uh, were exploring the California coast largely for furs from sea otters and other pinnipeds. Um, they also did scientific exploration, and that's when the first nudibranchs were discovered from the California coast. And so you can see that there's been a relatively slow progression and addition of other species through time, and with the biggest boon of describing the diversity of California's nudibranchs between 1950 and 2007. And the reason there was that huge increase was that we first developed scuba diving as an effective research tool to more effectively explore California coastline. And we're still finding new species today, and we'll talk a little bit more about that. But so much of California's nudibranch diversity has been uh, really discovered within our lifetime, and um, almost as much of the diversity that's been found in the history before that. So this period of exploration really, with the advent of scuba diving, brought us tremendous insights into the diversity of species on the California coast. Well, we still find new species, and here's two examples of things that we had never seen before that came to light in 2010. The one on top, this white species is found in deeper water in off of Carmel and Point Lobos area, and it was discovered by some divers who did some diving down to 120, 130 feet depth. And so it was a deeper water species that we just never seen before. Also in 2010, one of my colleagues, Jeff Goddard, who's studied a lot of the diversity along California's coast, um, found this new species in the tide pools of Santa Barbara. And so he sent it to me, and so I ended up naming it after Jeff, Paciphia goddardi. But we have a changing understanding of the diversity that we're seeing an increase in understanding that many of the species that we have traditionally recognized actually constitute more than one species. And in 2016, we found that the most common and widespread species in California, Hermescinda crassicornis, was actually two different species. And they have overlapping but distinct geographical ranges. Hermescinda opalescens was is generally found from the middle of Baja California all the way up to you find it up into Mendocino County for sure, but it's generally more of a southerly species. Hermescinda crassicornis which was actually named by these early Russian explorers, the same Russian colonials that established Fort Ross and several other places on the California coast, um, named this species Hermescinda crassicornis. And we found out usually genetic DNA data that there were these two different species. And so Hermescinda crassicornis is found from about from Marin County all the way up to the Gulf of Alaska. And so it is much more common to the north. Opalescens is much more common to the south. And the way in which you can immediately recognize Hermescinda crassicornis, the true Hermescinda crassicornis, the northern one, is that there is always a white line, an opaque white line that's present on the anterior side, the front side of the serrata, whereas Hermescinda opalescens uh, doesn't have that white line. And so for a long time, we just thought there was variation in terms of the color pattern. And then we found out that they were geographically and genetically distinct from each other. That was pretty surprising to all of us. And the same is true with this species, Limacea cockerelli, which was thought to be a single species all the way from Southern California up into Oregon and Washington. And when we looked at them, in this case, cockerelli was found from Monterey up north to Oregon and Washington, and Limacea macdonaldi was found from about Marin County down to Baja California. So again, the same pattern that we found in Hermesenda. So we're finding in almost every case where we really look at the California species in detail that there's more than one species. Sometimes there are more than two species. In some cases, there are three or four that have been masquerading as a single species. And so fundamentally, our understanding of the diversity of California is really changing, and there are many more species than we ever imagined. Probably the most extreme case is what was thought to be the single species called Doriopsilla albopunctata, um, again, being found from Baja California up to Oregon, is actually five different species. And probably the most common one you have on the Mendocino coast is this species we call Doriopsilla fulva. And so many of these are found in Southern California only. And there are two species, albopunctata and fulva, that make it to Central California and up into the Northern California as far north as Mendocino. And we're continuing to find new species that are not just um, species that are we thought represented a single species, but some that are just completely different, and we're only now recently discovering them. And these are two species that we have recently discovered in 2025. And this one with the bright yellow spots has a Native American tribal name, quack quack, which means yellow in the local language of the tribal group that lives in the San Diego area. This other one is found from from Southern California all the way up to Humboldt County, and it we called it Dodo Yurok, which means salmon um, in another tribal name in the Chumash tribal language. So recognizing them by their indigenous names is really a way of recognizing the heritage that so many, so much of what we know about California's biodiversity really comes from indigenous tribal peoples that were the original inhabitants of our shores, and we have so much to learn from that heritage as well as the new discoveries that we're now making. So I want to talk a little bit about human impacts and what's going on, and give you some examples that I know you're probably um, acutely familiar with, but just talk about those in the context of what we're recognizing and changes to the environment. We're finding, as new areas get developed, marinas are produced, and things that support recreation and fisheries. And we are seeing with the spread of California's population that there's a lot of habitat loss and reduction in the amount of suitable habitat available to it, our native species. We also find that introduced species create new biotic interactions, and I'll give you some examples of that as well, and really become competitors with a lot of our native species and place additional challenges on our native species. The other thing is we are experiencing a lot of short-term climatic shifts of warming waters, of cycles between El Nino and La Nina. And we talked about in as we were waiting everybody to enter this room that people had seen Hopkins rose nudibranchs on the Mendocino coast um, during 2014 when we had this pulse of warm water in 2013 and 2014. But once that warm water pulse disappeared, things basically returned to a lot of their normal situation. But then there are longer term climate change differences. And with warming oceans, we find that in the northern temperate region, that a lot of species are changing their ranges and are extending their ranges farther northward where there's cooler waters as the waters in the south heat up. Well, let's talk a little bit about the impact of introduced species. And here's three examples of a nudibranch that was first introduced to San Francisco Bay in 1982 from Japan. And it really pretty much just stayed in San Francisco Bay and didn't advance outside of the bay. In contrast, this other species that actually has an internal shell is found now in a lot of the embayments in California. It's found from San Diego all the way up to British Columbia. And it is a predator on um, clams that live in estuaries and mudflats. And so one of the things that we found that it feeds on the particular species of clams that are the primary food for a lot of wading birds like willets and curlews um, and it has actually caused the reduction of populations. And there is a lot of concern that it is really impacting the population numbers of some of our wading birds that are so important as residents in our estuaries and embayments. And then there's this species of bubble snail that has an internal shell. Um, that was introduced from Japan in the year 2000. And it also is spread from originally being found in Washington state and then making it all the way down to Central California. And one of the things that I mentioned how we use genetic tools like sequencing DNA. And this is how we've been able to firmly establish that these are species that have originated from these places, and that oftentimes there is um, genetic similarity with a particular population. For example, um, in Japan, where this snail is found, there's a lot more genetic variation than you find with the introduced population that's made its way to California. And when we look at the genetics of the ones that are in California, they're found in a particular part of Japan. And it happens to be the particular part of Japan where most oyster spat is sent around the world for growing Japanese oysters. So, undoubtedly, this species in this oyster farming region of Japan came on oysters as they were shipped around the world. And not only is it found in California, it's found in the Mediterranean, it's found in several places where oyster culture is a prominent way of um, establishing a successful fishery. So we know that a lot of these things have been transported by different mechanisms. The one in the middle here that came from New Zealand probably came as ballast water that was taken on in Auckland Harbor, New Zealand, and then discharged into San Francisco Bay. Um, and now, as a result of learning some of these lessons, there's regulation and laws in California that you cannot discharge ballast water um, in our embayments and near coastal waters, that you basically have to um, transfer, empty out the ballast water in the open ocean where these species can't survive, take on new ballast water, and then discharge that in the bay where you have open ocean species that can't survive in our embayments. And so this has dramatically reduced the new introduction of species here on the California coast. Well, one of the things that we're finding is that there are traditional boundaries of what we call different biogeographical provinces. So, from extending southward from the middle of Baja California all the way down to the coast of um, uh, to Ecuador, we have what's called the Panamic region, which is a tropical Eastern Pacific um, group of organisms. And then from the middle of Baja California up to Point Conception is what we call the Californian province. And then north of Point Conception is the Oregonian province. And those have thought to be relatively hard boundaries that you had a separation of basically tropical species, warm temperate species, and colder temperate species um, distributed along our coast. Well, I mentioned these cycles of El Nino and La Nina, and in 1983 we had a particularly strong El Nino, and the Spanish shawl nudibranch typically has this distribution from Baja California up to about Monterey Bay. But during the El Nino of 1983, this species made its way all the way up to British Columbia. It didn't persist as colder water moved back into these waters, but it gives us an idea of what some of the likely impacts of longer term climate change might be. And so, one of the things, for example, during El Nino conditions, when we did time counts of the numbers of nudibranchs that we were able to observe during El Nino conditions, there were a lot more individuals. On average, about 50 individuals that you'd see in about an hour of tide pooling, as opposed to La Nina, where it was roughly about half that amount. So El Nino conditions actually really create pulses of diversity, and the cycling of those um, really changes the population dynamics of not just nudibranchs, but all of the marine organisms that we find. So one of the things that we've seen with gradually warming water is that you have species that now have permanent shifts in their distribution. And so much like the things you would see during an El Nino event, um, these things have now become permanently established and are moving their way northward. So one of the things I've been doing since I uh, really started studying nudibranchs when I was in high school, and there's a picture of me in 1987 at the Bolinas Marine Station here on the Marin Coast, and we've been studying Duxbury Reef here in Marin for quite a few decades now, and counting. Um, that we've seen a lot of changes over time, and only because we have those historical records can we really get a good picture of what's going on. And we see that there are increasingly a number of species that are permanently moving their ranges northward. And one of the one species that is um, most complicated in terms of its increase is this species on the lower right called Phidiana hiltoni. And traditionally, if we go back in time from 1904 to 1975, the northern limit was Monterey Bay for this species. And then in the mid 1970s, we found that it made its way to Santa Cruz County on the northern side of Monterey Bay. And gradually over time, starting in about the mid 1990s, it made it all the way up across the Golden Gate to Marin County. And today, it's now moved all the way up to northern parts of Sonoma County. So this is an organism that you should be on the lookout because it is coming to a theater near you. And it has some impacts. One of the things that Fidiana hiltoni does, even though it's a native species, it tends to feed on other nudibranchs, and it doesn't feed on all nudibranchs, but it feeds on things, particularly the species of Hermacinda. And when Fidiana moves into an area, we find that there's a dramatic decrease in the number of Hermacinda, at least initially. And this is what we've been finding along the central California coast. And for example, if you look at the San Mateo coast, um, and you look at species that are not shown in gray that are not affected by Fidiana, doesn't feed on them, their populations have remained relatively stable. Um, but since the 1970s, when Fidiana first moved in, there was a much lower percentage of other species that it fed on, and those have actually increased over time. And because it's a relatively new arrival in Marin County, what we've seen is that there's a decline in these other species that are eaten by Fidiana. So it's a complex picture of change, but what we're now starting to see in Marin County is that there's a stabilization, that things are able to deal with Fidiana, that it populations are stabilizing, Fidianas in lower numbers, and the other nudibranchs that are fed on by Fidiana are making a comeback. And so this is a temporary destabilization of the populations, and eventually it comes back into more of a balance, and we're starting to see that as well. Well, climate change has also produced lots of unpredicted catastrophes. And one of the things that is nobody expected to see was the decline of starfish or sea stars on the California coast that occurred with the sea star wasting disease that really first started in 2013. We know from looking at bacteria that are present in um, specimens that are found in ancient collections that the bacterium is present um, in specimens that have been found in museum collections decades ago. But it's the warmer water that has really unleashed the fury of this bacterium and really caused the widespread wasting disease that we saw in 2013. And how many of you have seen a sunflower star in along the Mendocino coast since 2013? Has anybody seen one?
I think that in your audience tonight, there may be 3 or 4 people who've seen one or two. Actually, there's
Right.
a couple in our audience who've seen quite a few, but
And they're just beginning now to make a comeback. And there are profound consequences of particularly the decline of the sunflower star. The ochre star, which is common, much more common intertidally and feeds on mussels, has made a much more rapid um, uh, return to its original populations. But what we see um, in the case of the sunflower star, it's the primary species that feeds on the purple sea urchin. And with its disappearance, at least temporarily, there have been these huge blooms of purple sea urchins that you are acutely aware of on the Mendocino coast, and that has resulted in the decline of bull kelp populations, and the bull kelp has been in severe decline as well. We're seeing gradual return of sunflower stars. The Academy's Steinhardt Aquarium is working in collaboration with other aquaria up and down the California coast up into Washington. And we are rearing sunflower stars in these aquaria and with the hopes of re-establishing them on the California coast. Um, and the Greater Farallones Marine Association um, has been doing some transplant rearing bull kelp, removing urchins, and transplanting bull kelp back into kelp areas. And there are several populations along the Sonoma coast that have had implants of young kelp and removal of urchins and are really doing well. And so we can take this kind of drastic action to help restabilize those populations, and some of it will naturally occur anyway, but it will take much longer. So California's marine biodiversity is changing markedly. In our lifetimes, we have seen changes that probably haven't occurred for literally tens of thousands of years since the last ice ages. Um, so, what can we all do to protect California's marine diversity? We can certainly support the expansion of marine protected areas. The most important thing we can do to stabilize this situation is to shift towards carbon neutral energy sources and renewables. We can reduce other environmental stressors such as pollution and things like plastics and trash and other pollutants from um, industry. We can all become ocean advocates, and you are probably all ocean advocates that have joined other organizations such as the Noyo Center and are actively engaged in environmental action. And you are the eyes and the ears of the change that's going on in your tide pools by going out there and making observations and by posting some of those unusual things that you see and witness um, on iNaturalist and other platforms where scientists are then using that information. So by becoming community scientists and making observations, you are helping to document that change and be the early observers of things before they get out of hand. And we can take corrective action and things like iNaturalist and Beach Watch that the um, Greater Farallones National Seashore, sorry, um, the Farallones and Cordell Banks federally protected areas really help change all of that. And you are certainly really important key players in all of that, and particularly being located in more rural parts of California where it's harder to make those observations and there are fewer people doing it. So I want to thank you and I want to acknowledge support that I've had over the years from California Sea Grant and some of my colleagues, Jeff Goddard, John and Vicki Pierce, two amazing middle school teachers in the East Bay of the Bay Area, Bill Pence and Doug Mason, and Gary McDonald, who's one of my long-term colleagues. And all of you who make these observations and really help us understand what's going on in our marine environment. And so with that, I'd like to open it up to questions and basically see what things you're curious about and what I can expand upon and give you at least my best guess on several things. So thanks very much.
Thank you, Terry. Gosh, what you just said was so important, I think, for all of us to take in once again and just be reminded that getting out there and observing and noting your observations and the different ways that Terry was talking about, just so important and so good and so encouraging and so good for all of us. So excellent. I hope that this talk has inspired some questions. I know there's a lot of folks that get out into the tide pools that are here on the talk. And I see one question here. Where is the best place? It's a good one. Vivian is asking, where's the best place in the tide pools to find nudibranchs?
That's a great question. And basically, you can find nudibranchs almost anywhere, but the places that they really, where you can find a diversity is going out on a really minus tide in the lowest of the low tides, usually in the late spring and early summer months, which means getting up early because most of the tides at that time are just after the sun comes up. And so getting out there and um, trying to find your way out there in the fog sometimes is, but usually in fairly sheltered areas where there's um, protection from with offshore rocks that help shelter the tide pools where you are. Places like um, Stornetta um, is a great place to find relatively sheltered tide pools. And that's one of my favorite places on the Mendocino coast to go. But there are so many, and every little bit of habitat will have nudibranchs if you just spend your time looking. And it takes a little bit of experience um, to start to see them. And the more you practice and the more you go out there, the more you'll see and the more you will be amazed with the total diversity that's out there. But try and get an early morning low tide in the late spring or early summer, and that will be your best bet.
Nice. Thank you. Um, Barbara says, what strange and interesting creatures. And then let's see, Sheldon, who's out there a lot. Sheldon is I believe, retired high school biology teacher who I run into. Is that right, Sheldon? I run into Sheldon a lot, and I think he's got very keen eyes for nudibranchs. I've noticed. He's noticed urchins are feeding on sea stars and are now even eating sponges, and even with even animals with the nematocytes have you seen that as well?
Yes, urchins, prefer knife soft algae, but when you have an urchin baron, they'll basically eat almost anything. And in my experience, the only thing they don't eat is the pink encrusting coralline algae that um, is really too hard for them to scrape. But I suspect they even nibble at that. Um, when their preferred prey isn't there, they'll basically eat anything. And I've certainly seen a lot of that happening.
Mm. Yeah, the sea stars down where we go tide pooling often, you'll see the bat stars are just covered with urchins, and it's so sad to see because they're
Ah, yeah.
little, yeah. Um, yeah, and I think they even urchins will eat rocks. And then do you let's see, I want to make sure I'm getting all of these. Do nudibranchs bioluminescence here or abroad?
Yes, there are bioluminescent nudibranchs. Not a tremendous number. Most of them are found in the tropics. And when you touch them, they emit this really just amazing low intensity, bright green light. And it's really spectacular to see.
Wow. Is that because they're taking those bioluminescing critters into their bodies, or are they doing that themselves? Or is that, am I way wrong?
They're doing that themselves.
Okay. Okay.
Yeah.
Cool. Let's see. Is there a good field guide if we want a team of more of their, or let's see, if we want to learn more about their natural history, would you recommend a field guide?
Yeah, there is a really good field guide on the nudibranchs of California. Well, it's actually is species from Baja California all the way up to British Columbia. And the primary author on it is David Behrens, Behrenes,
Great.
ENS.
Yes.
Sorry.
Okay. Yeah. And I
Yeah.
wonder if we have that at the Noyo Center. We try to have good
And
field.
that's the most up to date
Oh, sweet.
one, and is the latest edition of it only came out a couple of years ago.
Oh, great. Good. Yeah, the life cycles would be really interesting to dive into because I only
Yeah.
started going, oh gosh, I saw a lot more little tiny nudibranchs in the spring. So yeah, lots and lots of questions. I better get read
Good.
some of these questions. I love it. You've inspired us. Let's
These are great questions, by the way.
yeah, really. Um, let's see. Let's see. Oh, you mentioned their teeth. And are their teeth, what are their teeth made of?
Their teeth are made out of the substance called chitin, which is the same substance that makes up the carapace of crabs and lobsters.
Great. Cool, cool. And that, you know what I love about that? One of our science teachers at the high school, I think, was trying to keep people from taking all those pockets of shells home because if you leave the shells on the beach, that they will go back and become those hard carapaces of the different
Yeah.
shells. It's a nice cycle to leave those shells on the beach. Let's see, in the local estuary, how do I find the invasive form from New Zealand?
Usually, if they're there, you'll see them just crawling on the surface of sandy, muddy areas right out in the open.
How big is that one from New Zealand? The like in the adult size, the
Yeah, so far I think uh, we've seen it fairly extensively in Tomales Bay and Bodega Harbor, um, and I think it's in Humboldt Bay, but I haven't seen it in a lot of places much north of Bodega Harbor, except for Humboldt Bay, and then as I said, it's made it up into Puget Sound and up into Washington, into British Columbia.
uh-huh. So we could easily see it
Yeah.
in this area.
But it's, it's, you know, uniformly white in color. It's found out in the open and it's pretty good size, about 2 to 3 inches in length. So if it's there, you'd probably see it.
Oh, okay. Great. Thanks for that. Let's see. Favorite tropical destination for nudibranch for nudie nerds on vacation. Did you mean nude nerd? No, nudie nerds. I know who you are. So,
Um, I I would say almost anywhere in the Pacific. You can see some amazing ones a bit closer to home by going to Hawaii, but my favorite places are in the Western Pacific, in the area called the Coral Triangle, which is made up of Indonesia, Papua New Guinea, and the Philippines. And the Philippines is my home away from home when I'm not here in California. And I'm usually looking for nudibranchs and studying them there, working with communities on conservation projects at least a couple times a year.
Wow, that sounds amazing. Um, so that's your favorite. And thank you, everyone. We learned new information, keeping our eyes peeled next time we go to the tide pools. Thank you. Absolutely. Can nudibranchs sting humans from their stinging cells that they ingest?
Um, they it's really an interesting process to watch them feeding on these things because, sea anemone, they still get stung by sea anemones. And so what you'll see them actually doing is sort of tapping their tentacles on the sea anemone and they'll get stung a little bit and it's sort of like eating hot chilies that basically, you have a little bit and then you build up your resistance to it and then they'll um, start taking a few chunks out of it. So they do get stung. I've actually seen some nudibranchs killed by sea anemones, but they weren't successful and the sea anemone won. But by and large, they will then incorporate those stinging cells in an unfired state, and then they transport them to the tips of their serrata, and then they're armed and ready to go.
Wild. Have you, I mean, you've seen it, and I just kind of just want to take that moment in because oftentimes when you go tide pooling, you're kind of like bouncing around from tide pool to try to see this one and that one and check it off your list. And I just am recognizing that to actually see that process that you've got to be laying on your belly and just watching one little area.
Exactly.
And that just what a
Yeah.
thing.
And when you're dealing with slugs, you have patience is the key to making good observations because it all happens in slow motion.
Right. Oh, I love that. Cool. Let's see. I want to make sure that I get to everybody's questions. Thank you for such an interesting presentation. Great photos, by the way. Um, and what are some key areas for research right now? Asking as an inspiring, as asking as an aspiring nudibranch PhD.
All
Um,
right. So well, there's so many different areas, and certainly understanding the genetic diversity. One of the really exciting areas is not only understanding what genetic differences are, but how various behaviors and things are related to the genetics and are differ in different groups of nudibranchs. And this is true of everything that we're learning so much about the genetic pathways and how different genes regulate certain activities and behavior. And I would say that's probably the greatest frontier out there for studying nudibranchs. But certainly even more mundane things such as using nudibranchs because collectively, even though there are lots of different species, and many of them are really specialists, nudibranchs feed on a wide variety of marine organisms. So if there are a lot of different species of nudibranchs that are present, that's telling you have a very diverse and healthy ecosystem. And so we're increasingly using them as model organisms to act as bioindicators of healthy or disturbed ecosystems. And so that's another ecological area that we're exploring a lot as well.
Great. Wonderful. How do nudibranchs get their colors? Is it It's from stuff they eat. Do they use some sort of process to convert it into bright colors? And how does the blue dragon nudibranch, which eats man of war jellyfish, catch them? Thank
Okay,
you.
those those are great questions. So nudibranchs get almost all their pigments from the food they eat, and they're able to change the chemical nature of the pigments so that they become more brightly colored. And you can find a nudibranch that will feed on a yellowish brown sponge, and it can produce pigments that are bright purple and yellows from different modifications to to those pigments. And they're the same kind of pigments. They're called carotenoids that make up carrots and things that we commonly think of as having bright colors, and they're able to modify the chemical makeup of those things to express different pigments.
Wow, that's amazing.
And that's another thing. How they do that, we have no idea.
Right.
So
Well,
there are many things for young folks to who are interested in them to study and really expand our knowledge and understanding of their evolution and how we can work for their conservation and their importance in global ecosystems.
exactly. I mean, it seems like we're just discovering new ones. And so there's the scratching the surface, and now how do they interact with each other, and how do they do this and that
Right.
and the other thing? Uh,
Yeah.
Amazing. Um, let's see. Squawk at me if I'm missing anything. And I see that the link to the book is in um,
In
the
the chat.
and the author is in the chat.
Right.
Link there. And again, the science talk will be online, or if you have any questions after the show, and you need to reach out to us, I'm sure there's contact information here for Terry. And oh, how long does it take for those eggs to hatch? So when you see a rosette,
Um, Usually, once the egg mass is deposited, in a tide pool or on a piece of algae. It usually takes between a week or two to hatch, and then the larval stages typically will spend another couple weeks swimming around in the plankton, and then when they detect their adult food, they then that triggers the presence of that um, some chemical from their adult foods stimulates their metamorphosis, and they immediately drop down to the bottom, crawl out, literally crawl out of their shell and become a nudibranch.
wow. I wonder if Henry knew that part. That is such a great question. I just learned something. Um, uh, Coming from a child's question and perspective, that's where we can do our best learning, at least for me. And Mary Glanville says here she's thought about taking a UV light out in the tide pool near dusk, wondering what would fluorescence. I know what you mean.
Yeah, and surprisingly, a lot of nudibranchs do fluoresce under UV light, and and so do a lot of the other um, organisms that, sea anemones often fluoresce, and and we think that this is sort of another kind of warning coloration, like having the bright colors, but that it's in the UV range that a lot of additional predators might not um, experience the same spectrum in our visible light range, and also rely on UV light.
And thank you all for being here. And we'll see you on our next few science talks. From little things that are magical to big things that are magical. Let's keep our heads in science, because it's amazing. We can get our minds blown. So thanks again, everyone, and have a great night.
Great to see you. Thank you again.
Thank you.